the lecture from last week, we went through the first 3 sections. And again, I'm not going to continue like totally go through them, but I will. So like there will be a question on these 3 layers of tissue that surround the kidney. And you need to know what is the function of the renal fascia, what is the function of the perirenal fat capsule, and what is the function of the fibrous capsule. Then we talked about the position of the kidneys, the internal anatomy, both as the like the larger structures like that cortex versus the medulla, as well as the structures that you would find in the cortex and structures that you would find in the medulla. So you do need to know where would you find things like the glomerulus or the renal corpuscle, which is the glomerulus plus the capsule that is in the cortex. The medulla is going to, this is where you're going to find like nephron loops. So looking for particular, okay. So you would need to know that you will find the glomerulus, the renal corpuscle in the cortex. You'll find the convoluted tubules in the cortex as well. And then in the medulla, you'll find the loop of Henle and the majority of the collecting duct as it runs down to the papilla. Know where things are located. That was mainly like the structure, the anatomy. You need to understand the blood flow. You might need to place that in order, and that would be on the arterial side. It's not going to be on the venous side, because the venous side has more ambiguity, I'll call it. So you do need to know the blood flow through the arterial side. This is something that you might see more in lab, where these are the glomerular capillaries, the glomerulus, the space, the capsule. A proximal convoluted tubule is fuzzy because it's the microvilli, and distal convoluted tubule is open. I haven't seen the lab exam, so I'm just guessing. I'm just reviewing. Who knows? We'll talk about the differences in cell types more when we get to acid-base balance and fluid balance. We'll mention them very briefly today. There are 2 types of nephrons. So which ones do you find where? You find the cortical nephrons are entirely in the almost entirely in the cortex. The juxtamedullary ones, these long nephron loops invade the medulla. They are going to be important in producing a more concentrated urine. Okay. So again, the capillaries. There are 3 types of capillary beds. We have the glomerular capillaries, which produce filtrate. And then we have 2 capillary beds that are absorbed, or their function is reabsorption. These peritubular capillaries are found in the cortex. They are going to be involved in reabsorption. The vasa recta are found in the medulla. They are also going to be involved in reabsorption. So know the 3 capillary beds where you would find them, as well as their function. The juxtaglomerular complex is here. It is comprised of 3 cell types. We're going to talk a little bit more about the role of the juxtaglomerular, sometimes it's called the complex or the apparatus today. There are 3 cell types. They work together to help to regulate. We're going to talk about glomerular filtration rate today, and that's directly proportional to the hydrostatic pressure. These juxtaglomerular cells are made up of 3 cell types. The granular cells in this afferent arteriole, these are smooth muscle cells that will vasoconstrict or vasodilate the afferent arteriole. The macula densa cells, this is the only place I've ever seen them called part of the ascending limb. In every other textbook, they'll be called part of the distal convoluted tubule. So that distinction won't be on the exam because it's a little bit varied, but these cells do monitor the salt concentration, because if the rate of filtrate flow is too fast, then salt can't get taken up out of it fast enough. So if the salt concentration is too high at the macula densa, then it will say, hey, wait a second, we need to be reabsorbing that sodium, and it will communicate with these granular cells to vasoconstrict that afferent arteriole and slow the rate of filtration down so that more sodium can be reabsorbed. The way these two populations communicate are through these mesangial cells. These have gap junctions. And so if there is, these are chemoreceptors, osmoreceptors. If they are getting, sensing higher or lower sodium concentration, that message is conveyed through these mesangial cells to the granular cells. So it keeps them all nicely clustered, but it also keeps them together for communication. This is leading into today that your kidneys will produce 180 liters of filtrate, but only 1.5 liters of urine are formed. If we didn't reclaim the water part of the filtrate, we would dehydrate in 30 minutes. So it's very important that we return 99.5% of the water, and that's going to vary a little bit depending on hydration status and blood pressure. But the vast majority of that is going to be returned to our circulatory system, or else we wouldn't survive very long. The kidneys do have a very high oxygen consumption rate, and this is going to be due to there's a lot of active transport. And the majority of that active transport is going to be of sodium. We'll be covering that today as well. So this is what we're going through today, the 3 steps of urine formation. So first, we'll talk about glomerular filtration. Second, we'll talk about tubular reabsorption, and then we'll talk about tubular secretion. With the loop of Henle, we're going to save the details of physiology for Wednesday, okay? So this is most of it, but not all of it. The first step is to produce filtrate. So we have, if you remember, there is an arcuate artery that is between the cortex and the medulla, and coming off of that arcuate artery are cortical radiate arteries. Cortical radiate arteries then are going to have branches off of them, which are afferent arterioles. So this is an arterial to capillaries to an arterial, so it's a type of a portal system-ish. But these are both, the afferent and efferent are both arterioles, so they are high pressure vessels. The other thing with the afferent versus efferent is the afferent arteriole has a larger diameter than the efferent arteriole, and this is going to create hydrostatic pressure. So pressure that pushes the plasma out of these capillaries and into the capsule, where it then will go to the proximal convoluted tubule. Glomerular filtration is a passive process. It is going to be dependent on pressures. Remember, so we're going back to hydrostatic pressure and osmotic pressure. Go to the whiteboard. For glomerular filtration pressure, we are looking at the forces that are pushing out of the capillary minus the forces that are pushing in to the capillary. For out of the capillary, we have hydrostatic pressure at the glomerular capsule. So this is called hydrostatic pressure glomerular capsule. This is created by the afferent arteriole bringing blood in, and then the efferent arteriole taking blood out, but at a smaller diameter, so slower flow. And that creates a pressure of about 55 millimeters of mercury. So we have our afferent coming in, then we have these glomerular capillaries and the efferent coming out. It's kind of like when you put coffee in your filter and then you close the lid and the water is forced through, and hopefully you don't get coffee grounds in your coffee, you just get coffee. So hydrostatic pressure is pushing fluid out. This is the hydrostatic pressure of the glomerular capillary, is pushing the fluid into the glomerular capsule. Then you have the glomerular capsule leading to the proximal convoluted tubule. Well, this filtrate has been pushed out and collected, but it's not under vacuum, like where the fluid's getting sucked out. So you do still have hydrostatic pressure of the glomerular, of the capsule. So I guess I need to figure out different, let me see which word you, I want to see what, there should be glomerular capillaries, and then, oh, capsular space. Sorry, I want to see which, what, so this is of the capsular space. And that is going to be pushing back into the capillaries. We also, if you use my coffee ground example, the plasma proteins, or those coffee grounds, do not escape from that filtration membrane. And so they are still in here. And that is going to be the osmotic pressure at the glomerular capillaries. Okay, so we have two pressures that are trying to force fluid back in to the capillaries, or in the case of osmotic pressure, they are pulling it back in. These are going to be negative pressures. We'll come back. I'm getting a little ahead of myself. Okay, so the hydrostatic pressure occurs because of the difference in diameter of the afferent arteriole and the efferent arteriole. That forces fluids and solutes, small solutes like glucose, amino acids, sodium, magnesium, potassium, through the filtration membrane. The filtration membrane, kind of like in the previous chapter, we had the membrane at the alveoli. Okay, so this filtration membrane is at the level of the glomerulus. It also has 3 layers. So instead of a respiratory membrane, we have a filtration membrane with 3 layers. Those 3 layers are going to be the fenestrated endothelium of the glomerular capillaries, plus a basement membrane, plus the podocytes. So just like before, you'll have to identify what are the three components of, this time it's the filtration membrane, from a list. And you'll have to check the three, which would be the fenestrated endothelium of the glomerular capillaries, the basement membrane, and these foot processes of the podocytes. So we have hydrostatic pressure of the glomerular capillaries pushing filtrate out through this fenestrated endothelium. It then diffuses across the basement membrane, and it will then make its way through these filtration slits of the podocytes. Again, filtration membrane. There are three layers. There is the glomerular endothelium, there is going to be a basement membrane, and then these foot processes of the podocytes. And these slits can be made closer together or further apart in order to either inhibit or assist in glomerular filtration. Okay, so major factor pushing in is hydrostatic pressure. Then there are, so this is the force, the pressure that is promoting filtrate formation. It's about, it's about 55 millimeters of mercury. Maintains its high resistance because most capillary beds have an arteriole and a venule. And so you get a drop in pressure along that capillary bed. But here we have an arteriole to an arteriole, so we can maintain this high hydrostatic pressure. The forces that are trying to push water back into the capillary, as far as hydrostatic pressure, or osmotic is the pull. These are the two forces that inhibit filtrate formation. So we have a negative 15 from the hydrostatic pressure of the capsular space, and a negative 30 from the osmotic pressure of the glomerular capillaries. So far we have three numbers. We have a positive number for hydrostatic pressure of 55 millimeters of mercury. So that is the driving force promoting filtrate formation. Then we have two opposing forces. These are going to be the hydrostatic pressure from the capillary space, because there is already fluid in here. So it's going to push back a little bit. The main opposing force are the plasma proteins that do not escape those glomerular capillaries. So just like in previous capillary beds that sucked water back in, these plasma proteins are going to also suck water back in. And we have a negative 30 millimeters of mercury. The forces opposing filtrate formation come to a negative 45. So that means that the driving pressure is going to be 10 millimeters of mercury. So there are four values: 55 millimeters of mercury. Sometimes this goes up a little higher. Negative 15, negative 30, and then a positive 10. You might need to match those to hydrostatic pressure at the glomerular capillaries, hydrostatic pressure at the capsular space, osmotic pressure at the glomerular capillaries, and then net filtration pressure. So again, more of those pressures. So we had hydrostatic pressure, 55 millimeters of mercury, that is the push. Then resisting it are 30 millimeters of those plasma proteins pulling fluid back in. Resistance from the hydrostatic pressure in the capsular space is 15. And net filtration pressure is going to have to be, and these are negative numbers when you use them. So the net force, net filtration pressure is 10 millimeters of mercury. This net filtration pressure determines the rate of glomerular filtration rate. If we decrease hydrostatic by even 18%, net filtration pressure goes down to 0. So it's important that we have a driving force that is significant. So this, again, is an excellent graphic with the numbers that you need to know. The only thing I guess I would change is that normally these are represented as negative pressures because they are in the opposite direction to hydrostatic. So hydrostatic pressure is a positive 55 millimeters of mercury. This can be adjusted up or down depending on if we are vasodilating or vasoconstricting the afferent arteriole. It is opposed by negative 30 millimeters for osmotic pressure, and then negative 15 for hydrostatic pressure for the filtrate that's already been formed and is sitting in this capsular space. Net filtration pressure is going to be 10. So you should be able to identify these numbers and what they could belong to. Hydrostatic pressure is always going to be the highest for the glomerular capillaries. Okay, so that push, it's got to be the largest number. This osmotic pressure usually stays pretty constant at 30, and that leaves the lowest one is hydrostatic of the capsular space. Also be able to calculate net filtration pressure. These numbers might not be exact. They may be off 1 or 2 so that I know that what you're actually doing rather than just measuring, memorizing 10. So if you're given 60, this would be 15. The glomerular filtration rate, the volume of filtrate that is formed per minute by both kidneys, is roughly 120 to 125 mils per minute. So this is usually pretty even between the 2 kidneys. So each kidney is roughly between 60 and 65. It is directly proportional. So the amount of filtrate produced is directly proportional to net filtration pressure. So again, if you go back to my coffee example, I can put my coffee grinds in my filter, and if I just pour water over it, then it just barely drips through. But if I put the lid on and it then creates pressure to force the water through, I get coffee, a lot more coffee, and a lot faster. So glomerular filtration rate, how much coffee we're producing, is directly proportional to the net filtration pressure. It is also influenced by the total surface area available for filtration. Just like alveoli, this is not normally the limiting factor, because surface area, unless it's a disease process, is usually sufficient. We don't limit ourselves by surface area. For glomerular filtration, it's really going to be net filtration pressure. How much is that hydrostatic pressure can we produce to create filtrate? Filtration membrane permeability, we can adjust this a small amount by looking at filtration slits. And then when this becomes damaged, this will be an indicator of kidney disease. So if the filtration membrane gets, like the endothelium or the podocytes get damaged and it becomes too leaky, which happens during renal disease, then we start getting things like proteins, not amino acids, but full proteins getting pushed out. It's like getting grounds in your coffee, which is not good, and indicates that something is wrong with that system. And we're going to talk more about renal disease at the next lecture. But big message from the slide is GFR, the most important factor is net filtration pressure. And the biggest regulator of net filtration pressure is hydrostatic pressure, because we're not really changing osmotic pressure. Those plasma proteins stay in there. And then the hydrostatic pressure of the capsular space is pretty consistent as well. So this is the main one, net filtration pressure. But the reason behind net filtration pressure is hydrostatic pressure of the glomerular capillaries. If with glomerular filtration rate, if the main indicator or main pressure is net filtration pressure, which is due to glomerular hydrostatic pressure, the way to control GFR is by controlling hydrostatic pressure. So there are two ways to affect. I love Starbucks too. So the main way to affect, and I'm just going to put this up here, it's really important, that what we're trying to change is the hydrostatic pressure at the glomerular capillaries. This is what we need to change in order to change GFR, because it changes net filtration pressure. There are two sets of controls, intrinsic and extrinsic. Intrinsic controls are going to dominate as long as mean arterial pressure is between 80 and 180 millimeters of mercury. So unless you're looking at blood loss or very high blood pressure, intrinsic controls are going to be in control. When we get outside of normal blood pressure, so when we get lower than 80, which is usually due to massive blood loss, or over 180, which is hypertension, high end of hypertension, then extrinsic controls are going to come into place. There are two intrinsic and two extrinsic controls. For intrinsic controls, there is myogenic regulation, autoregulation, and tubuloglomerular feedback. Myogenic is that we have our afferent arterial, and then it comes into the glomerular capillaries and it leaves via the efferent. We can alter the efferent diameter, but we much more often regulate the diameter of the afferent. So it's much more effective to, when you're looking at hydrostatic pressure, to monitor, to change the diameter of what's coming in versus what's leaving. So the myogenic mechanism involves the smooth muscle at the level of the afferent arteriole. When the afferent arteriole gets stretched, then it is going to vasoconstrict. And that is because it wants to keep a constant hydrostatic pressure. So if we start getting lots more blood flow, then glomerular filtration rate would go up a lot. So the afferent arteriole says, okay, we're going to slow that down. We're not going to allow as much blood to come through so that we can maintain a constant glomerular filtration rate. The kidney really likes to stay around that 60-65 mils per minute per kidney. So 120, 105 for both. If it can, it will auto-regulate to that level. So if blood pressure is increasing, then it causes the afferent arteriole to stretch, and the afferent arteriole is going to vasoconstrict in response. If the blood pressure going into the afferent arteriole is low, then those myogenic receptors are not getting stretched, and that'll cause vasodilation. Okay, so myogenic is the response of the afferent arteriole to stretch. For the tubuloglomerular system, you're like, oh no, another one. You already know this one. You've already heard this one. This is that juxtaglomerular complex or apparatus. So if GFR is too high, so if you have a high GFR, so you've got lots of filtrate being produced, it goes through those tubules really fast. And it's like there's a conveyor belt and you're trying to grab things off of it. I don't know, my niece, we play a Wii game and it's got like you build a robot. And like there's the easy version and it's very slow and you can pick the head and the body and the legs really easily. Well, she puts that on advanced and you're like trying to like grab body parts, to make these robots, and it's like crazy. So when the glomerular filtration rate gets too high, then it leaves salt. Instead of reabsorbing salt like it should, it is going to leave in the filtrate. The macular densa is at the distal convoluted tubule at the very beginning, and it's going to monitor sodium level. If the sodium level is too high, it means that the filtrate rate is too high for reabsorption. And so it is going to constrict the afferent arteriole to slow down the glomerular filtration rate and allow more time for reabsorption. So high sodium levels means that that filtrate is just flowing by way too fast and it needs to slow it down. So high sodium causes constriction of the afferent arteriole in order to lower net filtration pressure, lower glomerular filtration rate, and allow more time for sodium reabsorption. And the opposite would occur with low sodium. Okay. So those are the two intrinsic feedback mechanisms that happen during normal blood pressure, so 80 to 180. We have the myogenic, where if it gets, the afferent arteriole gets stretched, it's going to vasoconstrict. If there's not much stretch, it will vasodilate. But usually it's more on the end of it's getting stretched, and so it's going to vasoconstrict. We tend to have skew, like unless massive blood loss, we tend to skew more like to higher pressure. Like we put, like I get stressed, my blood pressure goes up, that afferent arteriole goes, whoa, what's happening now? And it will vasoconstrict to control that glomerular filtration rate. And then macula densa can go can be both of these occur, either low or high, but it's just easier to pick high and use that as an example. If we get out of normal range, so we get under 80, our mean arterial pressure of 80 or above 180, so our systemic blood pressure is way out of whack, we're going to start to sacrifice kidney function in order to increase blood pressure so that we have blood flowing to like our brain and our heart. Or we could cause it, if it's really high, then we don't want to burst blood vessels in those areas. We need to back it back down. So these extrinsic controls are going to prioritize the systemic system, the heart, the brain, the lungs, over the kidney. Because you can survive about 2 days with like no kidney function or suboptimal. So this is more, we need to take care of the brain and the heart right now. Outside of those levels, there are two mechanisms, neural and hormonal. We've gone over these actually a few times. So under abnormal conditions, the sympathetic nervous system will secrete norepinephrine and cause adrenal medulla epinephrine, so that sympathetic fight or flight response. And it is going to start to shut down the kidney filtration system. It says, I need that blood either somewhere else. That fight or flight, there's two factors. One is that the blood flow needs to be going somewhere else. So it needs to be going to, skeletal muscle. The other thing is it wants to shut down urine formation. If you are running from the bear, you do not need to stop and use the restroom. So those are the two things that are occurring with sympathetic nervous system secreting its hormones. It will vasoconstrict the afferent arterioles and slow down glomerular filtration rate. Low blood pressure is going to activate that renin angiotensin aldosterone mechanism, where under low arterial pressure, renin gets secreted. It's going to make angiotensin 1, which gets converted in the lungs to angiotensin 2, and then angiotensin 2 has those 4 effects. We've kind of gone over that, and so if you need a review, then you're welcome to watch. This is the linking to the same video we watched before. But the renin angiotensin aldosterone mechanism or system is going to result in increasing blood pressure when activated. So increased renin, cascade, 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 increases blood pressure. Whereas sympathetic stimulation is going to result in a decrease in urine formation and a shunting of blood to other systems. Okay. This is a very busy slide. I'm not really going to go over it, but because I don't really like it, but it does give you, it separates the intrinsic mechanisms that are active between 80 and 180 into myogenic and tubuloglomerular. Outside of regular blood pressure ranges, we get neural control by the sympathetic nervous system and hormonal control of the renin angiotensin aldosterone system. For the extrinsic mechanisms, because we've gone over them in other systems, these questions would be very general, if at all. We are currently studying the urinary system, so intrinsic control of glomerular filtration rate is something that you should study. You should understand myogenic and the juxtaglomerular apparatus. So assuming that we have now produced filtrate at the right amount and at the right rate, then the next step is tubular reabsorption. If we didn't reabsorb the water and the salt, then we would lose our blood volume in under an hour. So tubular reabsorption is a huge priority. There are two routes, transcellular or paracellular. Transcellular means it's going to go through the tubule cells. Paracellular means it's going to go between them. So this is the lumen of the tubule, and it is going to be called, this is called the apical surface. Then we have these tubular cells. The proximal convoluted tubule has lots of microvilli, so it has a huge amount of surface area. So transcellular, it's going to be, we'll call it transported for now, transported through the tubule and released onto, this is called the basolateral surface. For paracellular, it goes in between the cells. Normally these are tight junctions, but in the proximal convoluted tubule, they are leaky junctions. So trans is through the cell and para is between the cells. We're going to talk about which solutes get absorbed where and how they get transported. For the proximal convoluted tubule, it is going to reabsorb, where are my numbers here? So the PCT reabsorbs 100% glucose and amino acids. It is going to reabsorb 65 to 70% of water and salt. Also potassium, phosphate, and calcium. Okay. What else do we need to talk about? We're not going to talk about urea. We'll talk about that next time. Okay. So the proximal convoluted tubule is going to reabsorb 100% of glucose and amino acids, 65 to 70% of the water, salt, potassium, phosphate, and calcium. So how does it do this? The key player is going to be sodium. Sodium transport is predominantly through active transport. Okay, there is, in certain areas, there's going to be some diffusion, but we will talk about that with the loop of Henle next lecture. Right now, we're in the proximal convoluted tubule, but this will be applicable to other sections as well. But let's start at the beginning. Proximal convoluted tubule needs to reabsorb 65 to 70% of salt. It is going to do this by active transport, both primary active transport and then secondary active transport. It'll create the gradient for secondary active transport as well. If we look at, let's go to, okay, we're going to do this one. Okay, so this the sides are switched. This is now the lumen, apical membrane, basolateral membrane, peritubular capillary. At this basolateral membrane, so the membrane part that's closest to the capillary, there is a sodium-potassium ATP pump. It is very, very active. Okay, so that's one of the reasons why we get a very large consumption of ATP, is because in the tubular cells, sodium is getting actively transported into this interstitial fluid space, and then it gets sucked up into the peritubular capillary. Okay, so the first way is primary active transport. So using energy to take sodium out of the tubule cell and transport it essentially to the capillary. When it's doing this, so if we're taking sodium out of the tubular cell, is the sodium concentration going to get higher or lower?
The concentration in the interstitial fluid or in the tubular?
In the tubule cell.
Lower.
Lower, very good. So if we're removing it from the cell, it's going to get lower intracellularly. This creates a gradient for sodium to follow. So we pump it out to create a low intracellular sodium so that sodium will follow its concentration gradient and move into the cell. So we have primary active transport, and then we're going to get to secondary active transport as well. So the secondary active transport comes with, so now we've created a concentration gradient where we have high sodium in the filtrate, and it is going to move to this low sodium that we've created using primary transport. With co-transport, you can use, it's like you have a raft and you want the raft to travel on the river, but whatever is in that raft gets to travel too. So we can use this gradient of sodium rushing into the cell to take things with it. And so the things that we take with it are glucose and amino acids. So we drag the glucose and amino acids using transport proteins, but the energy will be from this ion gradient. So sodium transport, so in the proximal convoluted tubule, we need to move water back into the capillaries and sodium. We start with sodium for a few reasons. The way that we move sodium is by active transport, requires ATP. So the first part is this primary active transport with sodium ATP pumps. It pumps it out of the tubule cells and into the interstitial space that'll be adjacent to those peritubular capillaries. Then, because we now have a lower sodium concentration inside the cells, sodium will flow from the filtrate across that apical membrane, that's the one between the lumen and the kidney tubule, via facilitated diffusion, and that will pull co-transport other substances with it, such as glucose and amino acids. The other thing that happens is chloride is going to be attracted, and it will all be transported as well. And where sodium goes, water follows, and that's because of osmosis. So we have, I'm going to get a new one. So the first thing we're going to do is we need to reabsorb sodium. And we're going to do this through primary active transport using that sodium potassium ATPase pump to pump sodium out of the kidney tubule. That creates a facilitated diffusion gradient where we have ion channels that will allow sodium to come from the filtrate, now slide down that gradient into the cell. After we reabsorb sodium, oops. Okay. After we reabsorb sodium, we can co-transport with sodium things like glucose, amino acids, some other ions. Chloride will follow as well. Okay, so then we get co-transport. And because we've moved sodium, now water follows. So water follows sodium because of osmosis. So it all starts with sodium. You get sodium to move, then you can move other things. You get glucose and amino acids to co-transport with it, and you get water to follow because it creates a difference in osmotic conditions. Remember that isotonic, hypertonic, hypotonic? Okay, that's osmosis. You need to review it. So actively move sodium. Then you can passively move glucose, amino acids, and water.
I had a question.
Yes.
So is it the reabsorption of the sodium? Is it like steps? So the first step is the primary, and then the second step is the secondary, and then the osmotic water. Is that kind of how it does? I'm kind of confused.
Except for this sodium potassium pump is always going.
Okay.
So because it's always going, it's kind of that underlying foundation. It's always removing sodium. So it's always creating that gradient. Now, how much sodium would go in so this secondary process, this facilitated diffusion, would depend on how much sodium is in the lumen. But this pump is always cranking sodium out. So it is that foundation for everything else follows from it, but it's not sequential.
Okay, thank you.
Okay. Oops. So sodium reabsorption by primary active transport provides the energy and the means, meaning that gradient, for reabsorbing almost every other substance. So when we taught you about the sodium potassium ATPase pump back in 1141, this is one of the reasons, because it is in every cell membrane, but in this particular instance, the kidneys would not function without the sodium potassium ATPase pump. then after we establish this electrochemical gradient, we can co-transport glucose, amino acids, some other ions with sodium down its gradient. It's like grabbing somebody's hand and pulling them with you. Okay. The two main things that need to be reabsorbed are sodium and water. Without those, we cannot maintain extracellular fluid volume. So we've talked about sodium, active transport, then we get the facilitated diffusion gradient, co-transporting other molecules with it. For reabsorbing water, water can go through two pathways. It can go through that transcellular pathway if aquaporins are present. It can also go in that paracellular between and wiggle its way through. That's based on the osmotic gradient. So water reabsorption in the proximal convoluted tubule is facilitated in any and every way. Aquaporins are always present in the proximal convoluted tubule. And that is so that at least 65% of the filtrate water is reabsorbed, because we would die if it wasn't. So this takes priority, and so aquaporins are always present. Now the rest of that water, 30%, that's up for negotiation with the other requirements of your body. And so we'll talk about when we get to the collecting duct, aquaporins may or may not be present, and that's going to depend on hormonal intervention. But right now we're still in the proximal convoluted tubule. Aquaporins are plentiful, and so water will diffuse, osmose. So there are some things like fat soluble substances that they get reabsorbed and then they get actually resecreted because they're just going to travel according to their concentration gradient. The other thing that happens is that as water is reabsorbed, the solutes get more concentrated. So the salt gets more concentrated, the chloride gets more concentrated, and they're more likely to be reabsorbed as well. That one's not as important. So this is the, again, we're still in that proximal convoluted tubule, and it is going to show the processes again. So this first one creates that foundation. This is, okay, so this is the lumen. So the lumen keeps flipping back and forth. So always identify where the lumen is, because this is where the filtrate is coming through. And then we have our tubule cells. This is now the basolateral membrane because it's closest to the peritubular capillary. So that first process is active transport of sodium using the sodium potassium ATPase pump. That causes the concentration of sodium to decrease. So we end up with low sodium because of the pump. That creates this next step 2, which is facilitated diffusion. So it has to still go through ion channels, but now it's going from high to low along its concentration gradient. At the same time, we get co-transport of glucose and amino acids, some other ions and vitamins, that as sodium goes down its gradient, it basically holds hands with either glucose or amino acids, and it takes it into the cell along with sodium. So co-transport in the same direction, the symport. The two main things we need to reabsorb. So we do want to reabsorb glucose, we want to reabsorb amino acids, but in order for us to survive, we need to transport sodium and water. Water goes through aquaporins. It is going to follow its osmotic gradient. in and across to the cell. Lipid soluble substances, they will diffuse through, but they can also go back the other direction sometimes as well. And then there's this paracellular route, which is, we'll get into urea later, but also for water. This is a pretty good diagram. Again, it goes through all of the different routes for different substances. There is a transport maximum because sodium over here, this is a protein. Okay, so this is an ion channel or a channel that's going to facilitate glucose going through it. So these are protein channels, and it's like a doorway, and you can only fit so many people through a door at a time. Eventually, there is a maximum rate. So there is a maximum rate for sodium reabsorption. There is a maximum rate for glucose and amino acid reabsorption as well. Glucose is the one that tends to, and with like uncontrolled diabetes, will exceed that transport maximum. And then when we measure urine, one of the things we're looking for is glucose present in a healthy individual that's non-diabetic with healthy kidneys. There should be 0 glucose left in the urine. We'll talk about protein later. So there is a transport maximum. Proximal convoluted tubule, again, this is just re-summarizing that 100% of glucose and amino acids should be reabsorbed. 65 to 70% of sodium and water should be reabsorbed. Then you also get 80% of bicarbonate, which we'll talk about bicarbonate in the next chapter mainly. You get chloride, potassium reabsorption as well. And we'll also talk about urea later. So the main thing with proximal convoluted tubule is all of the glucose, all of the amino acids, and 65 to 70% of sodium and water should be reabsorbed at this section of the tubule. Then, we are out of time. Let me see. Okay, we just have 2 more slides. Okay, so then once we get to the nephron loop, we're going to go into this in much greater detail on the next lecture. But the descending limb, water can leave, sodium cannot. Ascending, sodium can leave, water cannot. We'll talk about that more. And then distal convoluted tubule and the collecting duct, you're looking at like 5% of sodium and like 8% in phosphate, and then the water and urea are going to be under hormonal control. So we'll go over this again next time. Okay, so this has what is absorbed where, and then we'll start back with tubular secretion. So we didn't quite get as far as I thought we would today, but pretty close. Okay, so I'm going to run class, and I will see you guys in 2 days.